this video we will discuss about this truncated O glycans in pancreatic cancer. So in the literature it has been found that in the pancreatic cancer there has been a lot of O glycan that are truncated. So this study decided to study the role of O glycans in detail on the pancreatic cancer. So what they did is they knock out this C1 GALT1 gene which is necessary for O glycosylation, and that study the effect of this pancreatic cells. And what they have find out is that when there is no C1 GALT1 mutations, the pancreatic cancer leads to a metastasis, and this metastasis is considered as a reference state. So, and in their study, when they knocked out this C1 GALT1, they can see that the increased rate of, they can see that the truncation of MUC-16 O glycosylation occurs, and also the increased rate of metastasis. So initially they hypothesis that the truncation of this MUC-16 O glycan leads to the metastasis of this pancreatic cells. However, they want to confirm this thing, and in their paper they have studied the role of this MUC-16 O glycosylation, and they have found out that in the case of this C1 GALT1 knockout, there is no there is we can say there is this MUC-16 O glycans, this truncated MUC-16 O glycans formation occurs, and that leads to the interaction of this truncated O glycans with this EGFR family and integrins, and thereby helps in the metastasis. That is being shown here. What it is does is it will help in this activation of downstream signaling pathways such as this PFAC and PAKT, and this will lead to this increased tumorigenicity and metastasis, and also the mesochemical phenotypes such as vimentin, snail, slug will increase compared to the case where there is no C1 GALT1 knockout. As you can see, there's in the case where C1 GALT1 is not knocked out, the MUC-16 is been MUC-16 which is a kind of O glycosylation is occurs normally. It is not in the truncated form, and thereby the interaction of this EGFR family and integrins is reduced in the case of this KPC mice, and it will not see the metastasis. It will not see the metastasis as aggressively as we can see in the case where C1 GALT1 is knocked out, and the data is shown in the panel. The data is shown in the figure on the left hand side. That is, when the C1 GALT1 is not knocked out, the metastasis occurs at the 30-week compared to the reference state, whereas when the C1 GALT1 is knocked out, we can see the metastasis at the 10 to 20 week in the case of this pancreatic cancers. So this not only indicates the role of O glycans towards this pancreatic cancer, however, it also provide the information that O glycans plays an important role in the case of this metastasis, which means the transfer of this tumor cells from one part of the body to the another part. And here we can see the different stages, or the we can see onset of these pancreatic cancers at different stages are shown here, and this aggressiveness of the pancreatic cancer has been found. Aggressiveness of this pancreatic cancer has been found to be more in the case of this C1 GALT1 knocked out, such as in the case of this pan-ion stage, which usually takes 5 to 15 weeks, will only takes 3 to 5 weeks in the case of the C1 GALT1 knockout. And that metastasis that I have already said, that metastasis will occur early in the case of C1 GALT1 knocked out compared to the case where C1 GALT1 is not knocked out, and the reason is shown on the figure on the right hand side, that is truncated MUC-16 O glycan helps in the interaction of this EGFR family and integrin receptors, and that triggers the downstream signaling pathways such as PFAC and PEKT that promotes metastasis and mesenchymal phenotype. That mesenchymal phenotypes means characteristic that helps the cells to move from one place to another place. So I hope you have understood or got an idea how this truncated O glycans promotes pancreatic cancer's metastasis. If you need to understand more about this role of N-glycans in pancreatic cancer, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.